Well, good morning or afternoon or whenever you're watching this. Welcome back to Year 11 Biology. We're going to have a little bit of a chat about cell structures. I'm going to set up some little vlogs regularly for you to check in with. And this is our first one. I'll get the PowerPoint up for us. So it's full. There we go. So this first one what we talked about in orientation week, but it's good for you to come back and refresh, especially when it comes down to exam time. So this is about cellular structure. So what's cell theory? Prokaryotes and eukaryotes. Limits to size, so surface area to volume ratio is important. Requirements of life. The cell's ultrastructure, which we'll talk about. You'll have to be doing some research for your assessment task over the holidays. And of course microscopes, how we find all this stuff out. Technology and science hand in hand. Okay, so cell theory. Well, there's three important guys involved in cell theory. Sorry, girls. Theodor Schwann, Matthias Schleiden, and Rudolf Virchow. And while they're generally recognised as the three guys that gave us cell theory, all living things are composed of cells. Living things, of course, are organisms. All cells come from pre-existing cells. Cells are the basic functional unit of living organisms, and therefore the cell is the smallest living organisational unit of an organism. So you can have a single cell organism or, like ourselves, trillions of cells. Reality is Schwann, Schleiden and Virchow used the knowledge of many other scientists and their own work to put together this theory. It's always a bit stunning for students of your generation to think this is something that wouldn't have been accepted. But we really didn't understand cells before this was all put together. So cell biology is really in some ways a newer science. Over 100 and something years old now. And of course, the rise of microscopes and the power of electron microscopes allowed us to really get to understand cells in much more detail, which we'll explore over the next 2 years. So cells form into two main groups. Life forms into two main groups, I suppose. These are above the kingdom level. The prokaryotes and the eukaryotes. And the prokaryotes are really ancient cells that predate the eukaryotes, and in fact there are some prokaryotic cells living within eukaryotic cells in what we call endosymbiosis, but more about that later. So what do they have in common? Well, they can be autotrophic or heterotrophic, so they can either gain their own food through photosynthesis or often more commonly chemosynthesis for these guys, or they're heterotrophic, they go and find food to eat in some way. So it may just be absorbing chemicals from the environment, but they're not making them, they're absorbing them. They're single-celled. They lack a membrane-bound nucleus, so their DNA is free-floating. And they don't have membrane-bound organelles. So it's all a bit of a slosh around inside the cytoplasm, which reduces their size. We'll talk more about size later. So they're very small, 0.5 to 10 micrometers in size, which is pretty small. The DNA is essentially one big solid loop, and you can see that pink thing in the diagram there, of chromosome, just one circular chromosome, and they often have lots of smaller circular plasmids which are also made of DNA. They can communicate with these and share DNA between cells using them, which is quite interesting and some new stuff that's been discovered. And their cell wall, so they have a cell wall, but unlike a plant cell wall, it actually contains a thing called peptoglycan. Suggests protein, glycan suggests glucose, so a mixture of the two. Excuse me. Now we're made of eukaryotic cells, so you find these in all other living things, plants, animals, fungus, and the protists, the other main groups within the kingdoms of life. And they share certain common characteristics. Again, they can be autotrophic or heterotrophic, thinking of plant cells particularly. They have a membrane-bound nucleus, which we can see in the diagram. It's a nice clear nucleus there. Nucleus there. That's a cellular membrane. And all these membrane-bound organelles as well. So there's lots more structures that have membranes, so lots more membrane required. And of course, all the DNA is inside here, the nucleus, and it's linear, so it's strings of DNA. And each species has a different number of those pieces of DNA. We have 46 pieces and 23 pairs. And the cells are much larger now. They're 30 to 150 micrometers. That's still pretty small, but much bigger than bacteria. So, cells are small. cells are small for a really good reason. They need to get stuff, it's a sort of single-celled organism here, they need to get stuff across this membrane into the cytoplasm. And that requires diffusion, generally, or osmosis, facilitated diffusion or active transport. But these are really determined by the speed of them is determined by concentration gradients, and reasonably slow, as we'll show in an experiment in class. So if you're too big, the ability to get stuff from out here that you might need, out here that you might need into here is limited, and the ability to get waste out of here out to the outside is also limited. So small is good. So therefore we talk about this thing of surface area to volume ratio. And so it's good to be small. There's that nucleus, pretend nucleus in this case, surrounded by some cytoplasm. Getting stuff from out here to the nucleus is going to be a lot easier here, a lot quicker than for this guy. So that smallness is quite useful. And cells are different sizes depending on their functions too. You'll hear a lot about structure and function this year and next. So this amount of surface area for a given volume tends to determine how big a cell can be. And the more surface area you've got, the better. So obviously, ways of stretching your cell out to create more surface area is also very useful. And it's another common theme we'll come across throughout the year. So, flogging a dead horse here, I know. This limits cell size. Limit is really about the speed with which most of the stuff it needs can diffuse in. Makes them small. Microscopically so, and we'll talk about microscopes now. So, what are these requirements of cells? Well, cells need oxygen, water, glucose, amino acids, and fats to enter on a regular basis. These are required for the cell's metabolism to create structures and the proteins which it uses. Cells also need carbon dioxide and metabolic wastes to leave because they're waste products, and proteins. They might be making proteins and getting them out of the cell. So they need to be able to get rid of them. Cells need to be able to reproduce, and cells need to be able to sense and respond to any change that might occur in the environment surrounding the cell or the environment within the cell. So just like the others, these are requirements of life really for all living things. So we talk about cell ultrastructure, and I left you with the task of discovering what all these organelles do, so I'm not going to do it here. Hopefully you've done that over the holidays and you've got yourself a nice PowerPoint you can keep updating. You'll need to update it. You'll learn more and more about each of them as we go, and you'll need to revise what you've got. But we'll point those out as we go along too. So we call these things organelles. They're small parts, cell parts within the organism that within the cell that perform certain functions. And each of them has a different structure. So again, we've got mitochondria here. I presume the mitochondria. Yep. In blue, that has a particular structure that allows it to do cellular respiration. We have a Golgi apparatus here, big folded membranes. These folded membranes over here that are rough endoplasmic reticulum and smooth, and all these other things. They're all certain sizes and shapes, allowed to fulfill their functions. And obviously, you all know that because you've gone and done your homework, I hope. So, how do we know all this? Well, yeah, these microscope things have been really useful. The Janssen brothers are probably those lens makers who are found to be responsible for the first microscopes. Robert Hooke is well known for describing cells by using a microscope, and a guy called Anton van Leeuwenhoek, a Dutch man who used lenses to check materials he was buying, was also known to have used a microscope, developed that idea, and probably used it more than Hooke did to explore ideas. He's the one who discovered that heat killed bacteria. He did that by checking what was on the scum on his teeth, basically, before or after coffee. So he got in the morning, scraped the scum off his teeth, looked on the microscope, and all these things were moving. But if he had his coffee first, they weren't. They sat still. They were dead. Pasteur used that idea later for pasteurization, which we still use, of course, in milk and wine and things. He also discovered that sperm are these little swimmy things, but we won't go into how he did that. But he's using a microscope, not that much like this one, but that's the general idea of an ocular lens and a oops, I'm going to use my finger. An ocular lens. With my mouse gone. Oops. This ocular here, and another lens there. And the two lenses allowed him to re-magnify the material, or what he was looking at. Of course, we put a structure around to hold it in place so he weren't, because Blue and Hook eventually was holding two lenses in his hands. So you can imagine how shaky that got. So the structure then became really important. And there's some lovely photographs of Robert Hooke's microscopes. Really quite beautiful pieces of building, of engineering. Stereo microscopes are a similar sort of idea, but in this case you've got two eyepieces. You often use them for things that are still alive, that you want to watch, so small bugs from the water, that sort of thing. And you want to have a look at how they react to things. In both cases, normally you'd have light coming from underneath, there we are, up through here. And so you need something that's really thin. You can use a stereo microscope with a light bouncing off the object, but generally, and that's quite effective as well, and you get a sense of where it's moving. All that heat, of course, tends to kill things off, so they don't stay alive for very long. But you're looking at surface, this is all about surface stuff. The light microscope gives you some internal features, but it wasn't until we got these guys that we really started to get a feel for what was happening around organelles and discovering what was happening inside the cell. Because obviously with cells so small, the cell organelles again are even smaller. And ribosomes defy description for a very long time because they're so tiny. But electron microscopes basically bounce light, beams of electrons, so really fine light, off a sample that's been coated in a heavy metal such as gold. So you're not getting a direct image, but you're getting an image that can be analysed and recreated as the structure that you're seeing. That's allowed us to see ribosomes and endoplasmic reticulum and mitochondria and chloroplasts and those sort of things in great detail and start to understand how they go about doing their work. So that's our first little session, and I'll see you soon in another one.